In my presentation, I will discuss which information from phenotyping can be used for breeding. The problem is that current breeding programs use genomic selection for yield and not trait-based selection or phenomics. So we can wonder what is the ecological niche of phenomics. The rationale of what I will present is that there is not one yield and one genetic control of yield when we deal with unfavorable conditions and climate change. So the idea for phenomics is probably to make use of alleles of adaptation with novel methods. When we look at a series of field experiments, the yield in these fields differs a lot between fields. The ranking of genotypes differs, and if we look at the effect of different regions in the genome, their effect of a given region is different between environmental scenarios. So for instance, it's not the same if it's cool or hot or water deficit or not. So there is no positive gene full stop, but genes that are best adapted to some conditions. So the role of phenomics cannot be to improve yield everywhere, because genomic predictions does better, but it's probably to identify and combine alleles of adaptive traits that are not used till now in breeding. And this is used for improving yield in unfavorable conditions. So how can we combine information of phenomics with field experiments? We can do that for identifying combination of alleles that best fit to local environments. So in one word, we can use data from the platform and here for the progression of the plant cycle. Then go to the field and calculate which was the flowering time or other phase of each genotype in each field. And if we can do that, then we can calculate response curve across sites, and we can calculate yield in all the fields. But with genomic prediction, we can do that in new genotypes. So see whether one given genotype will be favorable in Northern Europe, in Southwest France, or in Italy. And this is extremely useful for adapting genotypes to climate change. And so the number 5 is show that this prediction basically works even for genotypes known by their genome only.